not clear, but the outer layer will have the renal capsule. Let's focus on this field of view. You are able to see these dots. What structure is this? Yeah, it's the entire renal corpus because it has the Bowman's capsule and also the glomerulus. So these are all the renal corpuscles. That is the Bowman's capsule with the glomerulus. So if I am able to see this renal corpuscles, where am I? Am I in the cortex or in the medulla? Cortex. Medulla is almost all exclusively comprises of only the tubular portions, only the tubules. You don't see any glomeruli or corpuscles or any other stuff. So if you are able to see these renal corpuscles, then you are in the cortex part, the shell part. The field. Let us look at this field. Are you able to see any of those structures here? No. So you are in the medulla. What you are looking is the cross section taken through the medulla. You take one medullary pyramid. You have been looking at the longitudinal sections until now. When I take cross section, you see only circles, right? So this field is all the cross section of the tubules. We don't know, it might be loop of Fenley, some part of proximal column tubule, some the vasa recta, all other stuff, connecting ducts. All these are present in this field. But when I move to this field, I know that I am in the renal cortex. If I zoom in, I forgot to tell you one more thing. The point of entry of the blood vessels is called vascular pore, and the point where the glomerular fluid is formed, it's called urinary pore, like north and south pore, vascular pore and urinary pore. You don't see the other parts of the continuation of the Bowman's capsule, the proximal tubule, but you can see the parietal layer pretty well, pretty clear. Very flat cells, simple squamous epithelium. And the visceral layer, again, I said it's like a meshwork of capillaries. So it's again simple squamous epithelium, and also the visceral layer that will be intertwining along the capillaries. Pretty clear. I can see a flat layer of cells that's going towards the Bowman's capsule, and another layer, probably that's cut here, that's coming out. So which part is this? Vascular or urinary? It's the blood vessel. It's the capillary, the afferent arteriole. So it's the vascular pore. So roughly on the opposite side, we don't know, but we have the urinary pore. Let's look at this structure. This is the glomerulus, and here you see several tubules around. But you can see this tubule, it is not fuzzy inside. It's relatively smooth. But when you see this tubule, this long one, it's a bit fuzzy inside. Yeah, I'll zoom out so that you can get a better picture. I look at this tubule, it's more round and it's not fuzzy inside. And I look at this tubule, it's all fuzzy inside. Again, this tubule, fuzzy inside. Most of the tubule is fuzzy inside. When you look at this one, it's not fuzzy. So it's distal convoluted tubule. This is all fuzzy because you have the brush border. So this is the proximal convoluted tubule. Right? And probably this one that is really close to the glomerulus. And these cells are probably the macular density. It's not clear, but roughly in this region you have the macular density. I think this is the best picture. You can see these cells, clear lumen, so this is the distal convoluted tubule, and these cells that are close to the glomerulus are darkly stained, and on the other side they are relatively pale. So these darkly stained cells are the macular density. That's the reason they are called macular density, densely stained on histology. And if you happen to see an afferent arteriole, roughly there we might be seeing the juxtaglomerular cells, but that's not that. So this tubule, proximal or distal? That's distal. This one, proximal. Just remember, proximal it's relatively more long and more convoluted because it's the one that absorbs nearly 65% of the entire water part. So it's the most significant. And metabolically, say they are one of the most active cells in our human body because they have lots of cell membrane transporters that needs to transport all the glucose, amino acids, electrolytes like the sodium, potassium, chloride, bicarbonate, all that stuff. They are related. They are one of the most metabolically active cells in the entire body. Any questions in this slide? So you might be getting questions like in the field of view, you are able to see proximal convoluted tubule. But you won't be, I don't think we'll be asking questions like tip of the pointer because yeah, you just move it a little, solve the arrow. Maybe Dr. Vijay's class, if you take histology, she'll be asking tip of the pointer is on proximal convoluted or distal macular density. Now you are looking section taken to the entire kidney. Probably this is kidney of a monkey, so it's relatively small. Or I don't know if it's even a lower organ. But you can see the entire kidney. Yeah, here you are able to see the structure. What's that? The tip of the point. How does it look like? It has some stuff inside, so it's probably renal artery or a vein. It looks more like an artery. Maybe a segmental or the main artery. So the peripheral part of the kidney here you are able to see some corpuscles, renal corpuscles. This one. So this is all renal cortex. When I go more towards the center, here you can see all the longitudinal sections taken through the tubules. So medullary pyramids, and all these will come close and end into this space, roughly this space, the minor calyces, major and then. I wanted to show you the medullary veins. We know this is the cortex region, but we are able to see some tubules. These are the medullary veins. Generally, we divide the nephrons into cortical nephrons that are more inside the cortex and juxtamedullary nephrons. Juxta means close to medulla. So roughly here you have the medulla and some nephrons that are close to the medulla. They are called juxtamedullary nephrons, and these nephrons that lie in the cortex are cortical nephrons. Any questions with this slide? This is a slide. It's named kidney injected. So what the person who prepared this did is injected a dye into the blood, so that will go into the kidney, so it will be staining only the blood vessels and capillaries, and the rest of the part it appears more pale. So it's named kidney injected. So the pink part that you are seeing is almost all the dye, so that's the blood vessels. So what structure is this one? This big one. That's the glomerulus. Around the glomerulus you have the Bowman's capsule that's not stained that well. So this dyed part is the glomerulus. Another glomerulus, another one. And you see these straight tubules? Vasa recta. These straight tubules are the vasa recta. Or the peritubular capillaries. Okay, I am seeing a large blood vessel. I know that this is a blood vessel because it has dye. I see a large blood vessel. I know this is where you have the peritubular capillaries. It's all medullary pyramids, and this portion where you see the glomeruli is the cortex. What could be this blood vessel? The blood vessel that arches on top of the pyramids. Arcuate. That's the arcade. We don't know if it's the artery or the vein. Arcade, artery or vein. I'm thinking it's an arcade artery. So make sure you identify these structures, glomeruli or the renal corpuscles in the cortex, and the peritubular capillaries or the tubules in the medullary pyramids. Any questions on this slide? That's it for this week. We also have another slides regarding the urethra and all the structures, but urethra, it's again, what type of epithelium is urethra made of? Transitional epithelium. Just like the urinary bladder, the urethra is again made up of transitional epithelium. But I'll put the slide of the urinary bladder. you are looking section taken through the urinary bladder, you can see all the muscle here. Skeletal or smooth muscle? Isn't voiding under our control? Skeletal. So here you see the type of epithelium here, because the section was taken when the bladder was relaxed. When I mean relaxed, the bladder is not stretched. It's all folded and folded. So you see all these folds. And these folds, they are called rugae, just like the rugae of the stomach and the rugae of the gallbladder. You see these folds, rugae, and the type of epithelium. So the cells are arranged in more than one layer, in many layers. You can see the cells arranged in many layers. If the bladder was to be distended, these cells will be arranged in only a single layer. And the way that you recognize that this is transitional epithelium is seeing the cells on the surface. The cells on the surface are not flat. They are called dome cells, or they are dome shaped. Here you can recognize, you can see some cells that are not flat, some round nuclei. They kind of form like a dome. So this way you can recognize that this is transitional epithelium. If it was simple, if it was a stratified sperm epithelium, the cells would have been flat. Stratified cuboidal, all the layers would have been more cuboidal that way. But you can see the cells, they have something like a dome-like structure. They're not flat. They form domes, bumps. And again, underneath any epithelium, you have some connective tissue. Going down, you have the muscle, and on the outer side, you have the serosal curve, just like the intestines, same parts.